Ladies and gentlemen, it's my distinct honor to introduce Kevin Pullman. He is going to talk to us today about novel antimicrobial polymers for medical devices. And the reason I invited him to do so is not only because some people find this topic interesting, but because this young man is talking about commercializing it. And I thought with the shared love and attention of everyone in the group that we might have an idea or two that he might not have yet considered.
Thank you. That's absolutely spot on. So my name's Kevin Pullman. Company is HydroAir Global, and we are.
Say it slower.
What's that?
Say it slower.
HydroAir Global is the company,
Okay.
and the product that we are trying to commercialize we call polyhexidine, which is actually a combination of a polyolefin and chlorhexidine base. So I was actually kind of excited to see John in this discussion. I've supplied over the years, I've supplied Cook, at least for R&D, with some of the chlorhexidine salts, which is what people are most familiar with because those are the chlorhexidine compounds that go into solution. We are working with chlorhexidine base in combination with the polymer, and it's a homogeneous blend. So, you know, as was said, well, I'm here for two reasons. You know, I'm looking for a bit of input on the business side, and also, you know, looking for questions coming in from people that we maybe haven't answered yet. And we're trying to find opportunities that we maybe haven't thought of. So to give a little bit of background to where we are and the development work that was done so far, the origin of this product actually came out of a contract R&D project that was looking to do antimicrobials for water filtration applications. And although that project went in a different direction, this came out of that, and they had been doing polyethylene with the chlorhexidine in fibers for spun fiber water filters. And that was the early work on this. And the person who kind of took this up, his primary background was actually in fiber technology. He's ex-Kimberly-Clark, my partner. He ran one of the Kimberly-Clark R&D centers for fiber production. And most of his work has been based actually, you know, coming from that direction, looking at fiber-based applications. We've looked at some other things, but that's kind of brought us, you know, most of our beginning work has been fiber-based and particularly going into nonwovens.
Let me ask you a question or two just to set us up. First, a bit of trivia. You had Teleflex as a client of mine for a year or so, some years back now. And I remember that there were, I don't know if there were lawsuits or guidances about chlorhexidine, great concerns, recalls, deaths in Japan, I recall.
So yeah, I can address that. So with chlorhexidine, you had an FDA guidance put out, I think at this point, 4 years ago, for chlorhexidine gluconate containing products now have to carry an allergen warning.
Right. And for some reason it was accentuated in the Japanese.
Yeah, there seems to be a genetic risk in Japan, but the whole issue with chlorhexidine gluconate is a little bit unsettled science. I think people were content to slap the warning on the products and kind of move on. I keep looking, and I've tried to find people, you know, that work on this on the academic side to see if anybody's looked at it. It's kind of inconclusive, but from the people that I work with, and you know, just so people know, I mean, I've been working with chlorhexidine products now most of my career. My other business, you know, we represent one of the big chlorhexidine producers. So I'm in contact with a lot of, well, pretty much any of the customer base that have registered product in the US, and in discussion with people when this first came up, somebody whose opinion I respect, that really knows chlorhexidine products very well, his thinking on this is that it's not even the chlorhexidine itself that people are having the reaction to, that it is degradation products. because basically chlorhexidine is a really fragile molecule. It's sensitive to pretty much everything. And that's the problem for, chlorhexidine gluconate in terms of shelf life. Almost everything makes it fall apart. Heat, UV, regular light, uh, uh, radiation in general. I mean, it's sensitive to gamma as well. And that person had kind of hypothesized that, again, that what people are actually reacting to is, that companies may be doing things to their product, either in the sterilization process or just over the course of handling in terms of shelf life. And you're getting degradants that are, smaller fragments of that molecule that can penetrate deeper into the skin. But again, that's, still, I haven't seen anything definitive from a research standpoint. That sensitization and allergic reaction it's really very infrequent. And, a lot of what we saw here in the US, I think was triggered in the FDA warning. There was a lot of off-label use as well. So you have a couple of products on the market in terms of chlorhexidine wipes that have pretty specific label instructions, and it's being used in practice for a lot of things. And one of the things that it was being used for, and I think it's still used for, is on infants. And that's where some of the worst reactions were actually when it was used on infants in NICU. So for us though, again, we're not working with gluconate anyway. The chlorhexidine base is a much more stable molecule. And again, we're not even dealing with a liquid delivery type system for it to really penetrate the skin. And that's one of the benefits here that we have, and which I can get to.
That is my next question. Just quickly then, it was like an all hands on deck thing when I was there. So
Yep.
that's around the time of my engagement. People don't really talk about it anymore. So like, we know about that.
Yeah.
It's not
And I think part of it is, like anything that's a potential allergen, if the clinicians are at least aware that it's a potential, people are looking that it may happen and can deal with it. I think when it first came up, you're dealing with, anaphylaxis that happens when you're not expecting it is a much more difficult problem. So, those products just carry the warning now and people move on. But for Teleflex, part of the issue there is, their products, I assume that issue was on with the coated catheters.
Mhm.
Again, that's different from a general, external dermal exposure in any case.
Okay, which brings us to your product. So presently, your product doesn't exist commercially,
No,
and you are an engineer by trade.
I am not. My partner is the engineer. I am the business person.
Okay,
But my experience just on the practical side is, I've been working with people with, chlorhexidine-based products for 25 years.
okay, and there's an opportunity here because you've together invented something that doesn't exist, and the reason the community you're looking to talk to needs to know about it is because versus what they are presently using, this thing, what?
It is extremely powerful. So again, to put it in the context of where the original development path was, looking at woven and non-wovens, really for textile type applications. You look at other products that are on the market, whether they're outright coatings, a lot of cases for textiles wash-in. You have problems with downstream toxicity. And also, if you look just at what the label claims are for a lot of product, and it's particularly, applicable right now with COVID, which was also something, that kind of threw us off for, for this past year, because we were kind of ready to go last spring before COVID hit. And, I'll kind of get to it as far as the other, bacteria and things that we address. All of a sudden, the only thing anybody in the world cared about was, do you kill COVID?
And do you?
And we do, but we went through a cycle where we had to get new samples made, and then we had to queue up in line at the testing labs. We had our first test, we weren't really happy with the results. We had to go back and take a look. Is it an issue with protocol or is it our product? And it's basically a protocol issue and also time. So for most of what we can claim to kill, we get a very, very rapid kill compared to something like, again, on the textile side, the silver and zinc coatings, where their label claims are, that they'll get like a 3 or 4 log kill after, 4 hours. We get a 6 log kill in 60 seconds.
I remember.
So that's,
we talked about that. So
yeah,
let me clarify. Your product would be used in the preparation of a gown, a mask, a glove.
yes.
Okay. Anything that is worn, any kind of protective clothing.
Probably not glove. I don't think we can get it into material for glove. Again, it works with generally we've worked with polyolefins. It's PE, PP, playing around with nylons. And I can kind of go through. If I can do a screen share, I can even roll through here. But again, for us, the real benefit is that kill claim, and it's valuable in something like PPE where a lot of the infection in PPE comes with the practitioners touching their PPE as they're taking it off.
So I see you've done this, and now you're
yeah. So you're spreading it around the facility, and then you spread it onto yourself when you're trying to take it off. So what's a little bit confusing, and this kind of goes to the discussion we were having last week at the end with the proposed change in the rules from FDA, and on that list are things like anti-microbial masks and products like that.
Mm-hmm.
I'm interested to see where that goes, only because that guidance goes contrary to, or that change in rulemaking goes directly against the change in guidance that FDA had in the fall anyway, which kind of clamped down on a lot of those claims after they saw the influx of product coming in from overseas over the summer. But I can run through here.
Share your screen, and in the meanwhile, I'm going to leave a chat message for folks to remind them about last week's discussion about down classifying some relatively or highly risky things that FDA is thinking of not doing a full 510K review before giving clearance. We cannot see your screen yet.
I'm working.
Here we go.
There we go.
Okay.
So we're combining. It's a homogeneous blend of the polyolefin and the chlorhexidine. And again, chlorhexidine base is a solid crystalline product, and we introduce that to the polymer in the extrusion process. So we've done a lot of trials doing it into, we can do master batch polymer pellet, or we can go directly into the fiber. And we can, on the fiber side, we've done a lot to play around with the actual fiber characteristics. There are a lot of performance characteristics there, both from absorbency to just general feel of product. We can do a lot of things there in terms of multi-component fibers, different shaped fiber to affect its absorbency and things like that. And we also have done work, not as extensive, into molded parts and extruded film applications as well. But anything you can do with with polyethylene, you can do with this product as well.
How was your inventor come up with this?
As I said, this originally came out of a project to do water filtration, and they were the guy that did the original development work he's a polymer chemist down at Georgia Tech, who unfortunately passed away a few years ago. We wish we still had him, but he actually has pretty extensive research published into a lot of these types of products going into wound care, and has done quite a bit of work with chlorhexidine. So we've got 2 patents already. We have a few other extensions ready to go. And what this product forms is actually classified as a polymer alloy. So the chlorhexidine crystals are molecularly bonded to the polymer. And one of the benefits of that is that for everything that we've worked on so far, it is non-leaching. So that for some applications is a big benefit. Others, that's actually where I'm looking for feedback, particularly in the wound care side, which is one of the areas that made me interested in this project. I work with a lot of people with chlorhexidine in the wound care context, where things are going into hydrogels and things like that. We're looking at the base matrix here being antimicrobial. In some of the research, you see problems with the antimicrobial going into the wound and damaging skin at the cellular level. So, we need to find whether or not this is a benefit or not in the wound care space. And that's just a picture. We've done electron microscopy as we've gone through this. And again, one of the big benefits of this is, it can go in just like you can process it the way you do any other, polyethylene. The only thing that, takes a little bit of trial and error, as it does with any plastics process, is, you maybe need to tweak the system, particularly spinning fiber, looking at the extrusion process, but in general, it'll work like any other product. We think there's an opportunity, again, looking at some of the silver and metal ion based products that are on the market. EPA has kind of taken notice, and there's a potential problem for the silver based products long run, particularly the nano silver, because of what winds up in the wastewater stream. Again, our product doesn't leach out. From the regulatory side, we actually have interest from a lot of people in non-medical applications. The difficulty for us there is that opens us up. We actually need an EPA registration for that, which we are pursuing. I'm actually supporting a couple of companies that are dealing with active data call-ins for other chlorhexidine products with EPA right now. The only problem for us commercially with this is, number one, it's a lot of money, but more importantly, it takes actually a lot longer even than dealing with FDA, because you need both the federal EPA registration, and then after you have that, it's got to be registered in every state you want to market a product in with label claim. So that's a multi-year product, but there are a lot of people that have interest for spun fiber, for regular woven textile, as well as some of the non-wovens.
Lee, I hope that makes you feel better. There's something even more cumbersome than FDA.
Yeah, it's bad. I mean, we essentially have to allow for 3 years for EPA. We basically need 12 to 18 months for US EPA, and then another 12 to 18 months to get state registrations done.
I was hoping to get Rob Packard. I just wrote him on the side on the call because we worked on a project together that they went for EPA, and the way it had to be written and the like. If you have any trouble, you might want to talk to Rob. Please continue.
Yeah, well, and as usual, it's funny because I know when we first jumped on the call, when you mentioned he was saying he's working with the consultants, I tried to get a direct answer out of EPA a couple of months ago, and the answer I got back from the person at EPA was, go hire a consultant. They don't even want to answer the questions for you. So
I know a guy. What you took us past your curtains.
Well, that's one of the things that is an opportunity for us, mainly because that's been pretty well proven to be one of the, if not the biggest source of contamination spread in the hospitals. The barrier there is a claim on the curtains is most likely going to need also the EPA registration. So for us, the more immediate opportunities, believe it or not, are on the medical device side, under a 510K. So again, we think the opportunity is just that it is powerful, it is fast acting, doesn't add very much cost. We're in at a very low concentration. And again, we can play around with that by playing around with the actual fiber construction in any of these. We can really get our our component in the total weight of product down pretty low. And again, we're also working for water filtration as well as fluid filtration. We've had interest from some people for filtration applications on the medical side as well. So looking at data, again, the early data actually all kind of revolved around this drinking water application, and a lot of this testing there for particularly the anti-leaching was done to satisfy FDA, both under the AATCC testing as well as some protocols that were developed along with FDA to enable a drinking water claim.
Okay.
And we go through, and basically we originally, prior to COVID, we really thought that kind of the killer app for this in terms of claims was actually our efficacy against C Diff. C Diff is problematic and generally hard to kill, and it turns out that our product works really well on C Diff and as a general sporicide. We haven't tested directly on this product, but I worked on another project with chlorhexidine base where we also know that it's really pretty effective against Legionella. And here, if you look at our tests, we really loaded up the tests here with our challenge, and as I said, it kills pretty much everything right away.
Tell me about the speed and the comparison versus what's commercially available today, because I remember that it made me really pay attention when you told me the first time.
Yeah, so what we've seen for most other things, it's a little bit different if you look at, again, an actual device and going to something like John is working on. He can maybe speak to what they see looking at, either what he looked at an impregnated, but more significantly a coated type device, because there's been a lot of movement that way in that space with antimicrobial coatings. But for these textile type and fabric applications, whether woven or non-woven, everything that's on the market that we've seen so far has, again, these really extended time frames for their kill claims. 4 hours, 6 hours, 24 hours in a lot of cases, and that's particularly true for a lot of the product that's on the market with an EPA claim.
John, I know that you weren't on the engineering side, your mic is off. Can you talk to us at all about kill claims and how long it took, etc., back in your Cook days?
Speed is something that was out of my realm of expertise. Duration of the kill and the zone of inhibition was anecdotally 250 days. But for the regulations, I think it was a 60-day zone of inhibition, which was later dropped. I'm curious if this material can be impregnated with minocycline rifampin, or if that's not possible.
Probably not possible.
And it can be extruded in the catheter?
So we've done a trial. The problem that we've had with that application, and we haven't really had much in the way of resources to throw at that problem. So we work primarily with polyethylene, partly because of melt temperature, and we're trying to keep the temperature down on the chlorhexidine. And polyethylene doesn't have the flexibility, and the couple of trials that we ran trying to put elastomers in to make it more flexible kind of hurt the efficacy. And we think that what's happening is it's essentially kind of coating our product in the process. And we really haven't looked at it very much, honestly. But that's one of the things that is maybe a challenge for something like a catheter. It's really trying to figure out how to get it into a product that has the flexibility.
My Joe Bag of Donuts perspective is such that C Diff is huge. I don't think there's anything on the market that prevents that, and I'm not sure it's an application that would fit. But would the woven material have use as a endovascular stent graft woven material, or is that too fine of a material that would make it impossible to use?
I don't know, and maybe we can have a discussion offline, only because I don't know. But what's your question? That we need to get it to be extremely fine?
Yeah, and it may be a solution in search of a problem, but I know that endovascular stent grafts have loomed material that are a key component, kind of commodity space in terms of all companies use that.
Yeah.
I just know that the Spectrum catheter was astonishing in its ability to. It was virucidal, broke down cell walls. There was, though, an embedded resistance to it at the hospital level because there were individuals who simply believed if we could get physicians to wash their hands, this problem would go away.
Yeah.
Despite all the evidence over the last decade that it doesn't work that way. It doesn't work. Washing your hand is wonderful, and bundled sterile approaches are wonderful, but mysteriously, pathogens still colonize, and people still die at an astonishing rate. Every night an airplane goes down in America, and 60% of the people on board die of a catheter-related bloodstream infection.
Yeah.
And everybody shrugs it off. It's an astonishing level of mortality that is in real need of a solution.
Yeah, I mean, I think for us, from the fiber standpoint, what we draw for these other fiber applications is extremely fine denier fiber as it is, and our most recent process development was doing the multi-component with a polypropylene core and our product on the outside. And we can certainly play around with that. I think that's possible. So, I would be interested in a follow-up conversation to get your thoughts on that.
I've been gainfully, voluntarily retired from Cook, but I'd certainly help you negotiate the labyrinth of Cook if you need some suggestions on
Sure.
who and what and why. And Joe, I'm extremely uncomfortable with my head on this screen, so
It's just
you, Kevin. Thank you.
it's
You're
just so big.
Yeah, it is prodigious.
You have to, you need to put the photo of you as a child, which you trot out from time to time on Facebook. It is just the largest unfortunate childhood.
There's a lot of synapses up there shooting around, I mean, you need to have a cavity large enough to hold them all, I think.
It's funny. Eddie,
So this just flipping through here quickly. So this other test, this is a challenging against staph, and here we had a control sample against an off-the-shelf surgical mask.
sorry, Kevin, you don't realize that you're not presently sharing your screen.
Okay, hold on.
Go ahead and do that.
Uh,
Hey, Eddie.
go back. All right, we're back.
We are.
So this is what we got against staph, against a standard this is a Kimberly-Clark mask. They actually coat their masks with a kind of a mild coating, and that's kind of give you a comparative idea what it looks like on a mask. And again, with all of these, we went with extremely high challenges. We should get into this. This was a catch-all early on when my partner was trying to get this going. He didn't have the money to test everything independently, and they basically made a soup of an awful lot of stuff. And again, it kills pretty much everything. So our problem when we got to COVID was that our first test, we actually tried to do the same way we did our other tests, and we tested for a minute, and didn't work so well. And there were some other things going on in the protocol that we had to look at. So we went and retested at 15 minutes. We basically get 1.6 log at 15 minutes. We think actually if we extend that to 30 minutes, we'll probably be up at 4 log. And again, the products that we've seen that have come on the market for that have been approved with a claim against COVID are typically listed with FDA at 4 hours or 6 hours or 10 hours. So we still should have. All right. So I see questions from Dwayne. We are right now kind of getting to the biocompatibility. That's kind of where we are in our development phase.
I'll bring him on in a moment.
Yep. And quite frankly, that's something we haven't done yet. What we have though, which kind of encourages me is, again, we're dealing with chlorhexidine, which is kind of a known entity, and the biocompatibility is not coming, completely out of left field here. So we're really trying to start down that path. And part of our issue is that up until now, we really weren't sure kind of what the first products were going to be and what the requirements were going to be for that product. We, out of necessity, we have to prioritize, and we just didn't want to kind of go crazy with testing for testing's sake. But we're probably going to get started with some very basic biocompatibility and sensitivity screens.
Real quick, Dwayne, for the benefit of those who did not read your question, what was the point that you made?
Oh, that was funny, but I was saying, great, we understand it kills everything, but if I'm going to be a buyer of this, what's the biocompatibility? And then just as a side question, I was just feverishly reading some papers on my other computer here about this. I see a lot of comparison studies with Alexidine. I'm sure you're familiar with this. Is that a competing product, or?
Not really for these. I don't think you can get it into the product the same way.
Yeah. Okay. Yeah, I was just curious. I saw some research out of like Case Western and.
Yeah, I mean, you've got a couple of other products that are looking to compete with in some of the liquid-based like the surgical scrubs and things like that. Some of the other bisguanides. So you got Alexidine and octenadine. Part of the issue with those, and honestly, I'm working with octenadine on a couple of projects. And for that, you have the barrier that from, at least from FDA's standpoint on octenadine, it's a novel drug. And that becomes a giant barrier to getting it into any product.
Yeah, right.
And I haven't seen if Alexidine has been registered in anything here yet, but octenadine, it's on, like octenadine's on the market in Germany, and that's pretty much it. It hasn't really even taken hold through the rest of Europe, even with some, semi-harmonization across Europe. So I think it's going to take a while.
Okay. Yeah. And with the biocomp, because this is my area, I'm curious because, whoever ends up using this product is going to have to rerun biocomp on their actual device, right? But how do you alleviate the risk for them of using something new? Just as a suggestion, I know you did a leaching test. I'm curious to know, the parameters you ran that under. What I would suggest, if it wasn't this, is running it how chemical characterization would be run or extractables leachables will be run on a medical device, right? So using water as a solvent, ethanol and hexane, just to truly make sure that there is nothing leaching out, because those are that's the environment that
Yeah.
a medical device team will have to run it through. So just a suggestion. Happy to talk offline, but
Yeah, I appreciate that. Yeah, and that's what we're getting to. I mean, for those previous studies, I mean, it was all based on water. The one that I'm most interested in seeing is probably the hexane.
Sure.
And seeing, and again, I'm not even necessarily that concerned about the chlorhexidine itself. The issue with chlorhexidine in general is, again, the impurities. And I'm curious to see on those tests how the hexane affects the impurities. It probably should be okay, but as with anything else, it probably is then also dependent on the quality of the chlorhexidine itself as a starting material, because, and that's something that's been an issue for chlorhexidine in general over the last 25 years. In the traditional applications, FDA has taken, a closer and closer look at elemental impurities and degradant products and things like that. So, we've worked really hard to get the total level of impurities in the base down, because that's what affects, the impurities in any of the salts.
Yeah. And I think, with the hexane too, if you do get poor results, it's very easy to get that kind of data thrown out, because it's very clear from the FDA that they want you to challenge the device, not degrade it, right? So
Yeah.
Interesting, very cool stuff. I'll shut up now.
I want to introduce Ed Lin. I typically, as you all know, only promote folks on screen that are full MDG premium members, but Eddie's a special case, and he's always kind of been a special case in a lot of ways. But Ed has been working on a wound care device for, well, a really, really long time. And
prehistoric, Joe.
prehistoric.
Yes.
You were talking earlier, Kevin, about its potential application in wound care. So I wrote him on the side. I was like, you got to get on this call. Start anywhere. Kevin, say a thing about wound care. Ed, say a thing about what you're doing, and let's see where the two mix, if at all.
Well, Kevin, because I joined belatedly, so I didn't hear all your presentation, but my understanding is that you have a new chemical, probably in some ways
old chemical in a new application.
Okay, all right, yes. That is antimicrobial and bonds to non-woven fibers. And we are working on dressings that have active fluid transfer properties that will selectively and actively move fluid from a wound bed away from it in such a way that it would not cause any kind of skin maceration. And the benefit of that in negative pressure wound therapy is that then you no longer have to trim the dressing to the exact geometry of the wound bed.
Yeah.
And we have a therapeutic platform that also additionally delivers alternating cycles of topical pressurized oxygen. So currently, all negative pressure wound therapies relieve their suction by using room air. We use pure oxygen or therapeutic fluid to relieve that suction pressure. So in so doing, that adds 8 extra hours of beneficial therapy to the patient each day that didn't exist before.
Yeah.
And we also recently just received the go-ahead from the FDA to market a special mask system that can be powered by low flow oxygen, such as only 5 liters per minute from an oxygen concentrator, but delivers extremely high, 80% or more FiO2 deep into the lungs of patients. And that's something that has not been achievable before, except by using a ventilator. So our system can use filters that would either filter the virus going into someone if they are trying to, prevent themselves from getting COVID, or protect other people working near the COVID patient from getting infected. So that would have, important applications in the military, where servicemen they have mission critical responsibilities. They are asymptomatic, then they can continue to work next to other service members who are not infected. So I see some possibilities of cross application, if you will.
Yeah, yeah. And for some of those, I mean, you've got the direct face mask, but looking at some of your other applications, there are possibilities for filtration, either with a non-woven substrate, or you can go with something like a sintered polymer as well, particularly if it's low flow.
Yes. Great.
But I'd welcome a follow-up conversation.
Okay.
Eddie, again, my knowledge of all this being so cursory, I just heard the magic words of wound care. I thought Ed Lin. Did you hear something from Kevin just now where you're using one set of chemicals that this might be a better solution than what you're doing?
For our immediate application, which is to provide very high oxygenation to a COVID patient, we did not have consideration for a antimicrobial impregnated filter. We have considered using antiviral filters, but they are not impregnated with any antimicrobial.
And the delta for you, would you believe be significant, or that's nice, but not on a critical path?
I think it would have use for people who want to either avoid catching any residual COVID viral particles that might get through their, 97, 98, or even 99% viral effectiveness filter. Now, it is something that can be added on to make it perform at a even higher level. But the issue is, of course, cost and how long it would take to get FDA clearance to add that to the existing viral filters.
Well, I don't know what Kevin knows about how long they'll take for FDA, but you probably have a sense of cost. Kev?
Well, that's hard to say because that's dependent on the device. Again, as was pointed out by Dwayne, it's the finished device that has to get tested and approved ultimately.
Mm-hmm.
So in this case, the finished device will be a viral filter that has a diameter of probably, let's say, 5 or 6 centimeters that fits in anesthesia circuits or respiratory circuits. There are existing manufacturers of this, and you would have to, interest them to add your antimicrobial to the filter.
I may actually have somebody that is partway down that path that's done some technical work that we can maybe connect with.
Okay. Well, this is it will not be easy.
Yep.
Let me just warn you of that. And the people who would have the money to afford that would typically be the military.
Yeah.
Because they don't care too much about cost.
Yeah. So, well, I mean, that the question there is, do you have connections with those people? Because to get into that pathway as an unapproved vendor to the military is a challenge, because that's something that we looked at already last June when DOD had, all of these projects going out there to try to source, masks and things like that. And if you're not already an approved military vendor, it becomes much, much more difficult to get in.
You're correct, Kevin.
Okay. Thank you, Eddie. And Kevin, I've already given you Ed's email address in Slack.
Thank you. So I just got one or two more slides here.
Oh, okay. Let's have you put them back on, because once we were just talking, just a bunch of talking heads, I made us bigger and got rid of your
Let
slides.
me put it up here. I don't know why it's not coming up on my.
In the meanwhile, Carrie, you want to slip in with your question? And ask you to start your video.
I just wanted to throw up the C Diff. If people
Okay.
were interested in that.
Sure.
Hi, yeah.
Oh, just a sec now. We're going to talk about
Eric.
C Diff.
Oh, sorry.
All right. Stay where you are and put on your camera if you like. Go
I'm
ahead.
in my jammies. I'm not sure if I should.
Oh,
All right, well, let's give it a go and we'll see. Good morning.
it was a great
Good
jam.
They're having my coffee.
Kevin, go ahead and tell us about C Diff.
No, so I just wanted to put the past just again, C Diff is typically pretty problematic, and you know, on this, it's really effective as well. So you know, we thought early on that you know, there were also opportunities in you know adult incontinence care, particularly for the nursing market, you know, to try to address some of the C Diff issues. And this is another C Diff test. So we think actually that one that second test there, we think there was actually an issue with our sample.
Thank you for that. I think that's the end of your slides.
Pretty much, unless anybody wants to get into how nonwovens are made. So
I would say among the folks on the call, maybe Jorg is interested, and he can let me know if he is. Other than that, I can, you know, we can be sure to get this presentation.
yeah, that's pretty mechanical process.
Okay. So yeah, be sure to share the slides with me, and I'll make them
Sure.
available for the group. Okay. Put John
So-
Lady.
Carrie.
Oh, hi, yeah. So I make a little product that's made of silicone, and sometimes it gets damp and you carry it around and you want to put it in a little bag, but you don't want it to stay, you know, moist. When I was looking at this, and I came in kind of late and I was just looking at those curtains, would this be a polymer that would work to make little cases for dentures or retainers or things that you want to carry with you for a period of time that might be damp or have germs on it? That
Yes.
like bathing suits when you're traveling, or just different things that you need to keep clean for, that you don't want to start growing mold in the half hour or
Yeah.
6 hours while you travel.
So for us, when we're looking, that's one of the things that we're looking at in those, you know, an item like a molded item like that with a smooth surface. The issue that we have is again, you know, it's one of the downside to the fact that it doesn't leach is you're kind of incumbent on whatever is possible contaminant touching it and getting good surface contact. So, you know, for the fabric side, we can play around again with fiber shape to basically, you know, massively expand surface areas and to increase absorbance. It's harder with a solid surface product. We've played around a bit with, particularly with films on surface modification to basically break surface tension where it works.
Mm-hmm.
We for something like like that kind of case, we just haven't had time to play with it. But there are things that can be done with surface modification that you know would allow that to work.
Yeah, because I could picture, I mean, and I don't even know if this is an application for it, but you know, when you buy a toothbrush holder or you buy something, you're looking at it going, why would this be good to put my toothbrush in?
Yeah.
You know, in a closed environment that has no antimicrobial properties.
Yeah. So the issue for us with those applications though is again, it gets us back to EPA registrations, which take quite a while. Takes a long time and a lot of money. And it also comes down then to what exactly do you want to have the claim for. So something like you're looking for is actually the easiest claim to get with EPA, and will actually be our starting point for registration, and that's what they call a treated articles claim. which, you're basically saying you're keeping that article from getting moldy or contaminated with whatever. That's without a direct human health claim. You aren't saying that, we kill, staff or whatever. You're just saying we're protecting the product itself from becoming contaminated with whatever.
Did you
but it's
talk
gonna
about
take-
with food with food items?
Yeah, actually, again, since we started this whole discussion with FDA for drinking water applications, we had also discussed with them food contact, and we would need to do some additional testing, but based on the initial testing, we should pass, but we would have to do a registration for that. And again, we certainly can do things like films. We can do extruded films. We can essentially make a saran wrap type product with these claims, or something like a sandwich bag or anything like that. What's that?
This product would it do anything to keep mold from growing on cheese?
I don't know. I thought you want mold on a lot of cheese.
I know, but a lot of cheese would, how you put vinegar in with so I'm thinking, God, wouldn't it be good to have a case that would stop all growth of mold once you get to that taste you like?
Yeah. That those sorts of things start becoming though commercial questions, and what are, given the regulatory work that has to be done to support those claims, how big is that market, and what are people willing to pay? So
from Dave that says that there are antimicrobials already on the market for plastic articles such as Microban.
Yep. Well, Microban itself is, nowadays it's not one product. Microban is a company, and essentially they market the Microban name for a lot of different products. And again, it depends on what kind of application you're going for, but it also comes down to what kill claim do you need? And if you look again, for Microban, they have some products in the medical device space, and offhand, I can't remember which chemistry they're using there. It's much more familiar to people in the consumer space. Over the years, they've actually gotten in trouble for some of their claims. They actually got bounced off the market for a while when they were marketing off the Microban cutting boards and things like that. But depending on the product that it's going into, it's different chemicals. And in a lot of cases, it's not even their own product. They're sub-registering EPA registered product from other people that they then market under the Microban banner. And the flip side to that is, I think the big one for this past year is you've got the Microban hard surface sanitizer that was it's marketed now by P&G. P&G snapped that up just in time for COVID to hit, and they've been pushing that Microban 24-hour hard surface sanitizer pretty hard this year. But for Microban, and there are other products out there, but we think that for some of the spaces where we have applications, just the fact that we're dealing with chlorhexidine in the medical space, it's a very familiar product. You're not coming in with totally new chemistry that will be a barrier both for the customers as well as for the regulatory authorities.
Gary, before you're both on Slack, so you can follow up with one another. What polymers, if any, are you using today?
Primarily polyethylene.
Gary, for her
Oh, sorry.
Oh, I'm using 100% silicone. Yeah, just FDA approved silicone, and so I don't know if there's a possibility to mix any of your antimicrobial.
Yeah, I don't think so. I think we get back with silicones. I think we run into that same problem that we had with trying to blend in elastomers, where I think the silicone would wind up kind of coating it. So again, the basic technology we have is again forming that molecular bond with a polyolefin, and that won't necessarily happen with other molecules.
Well, yeah, I mean, I was thinking about, a friend of mine is doing an invention for a medical device, it's a class 1 medical device that simply helps people who have very limited mobility pull up their pants.
Yeah.
But it needs to be almost like just like fabric elastic, but it has to have that antimicrobial property,
Yeah.
Microban, I don't know why that name is familiar to me, and she even mentioned it, but it wasn't suitable for her, what she wanted to do. So, I mean, there's so many applications for it.
Yeah. So for us, it's a combination of, does the polymer fit the application?
Yeah.
And for us, again, one of those benefits, and you look at Microban. So depending on the product that Microban is selling, they have some that are impregnated into the products, and a lot of what Microban does is also coatings. And for us, on the textile side, a lot of what we see as the competition are coatings that come off over time. So
Yeah.
it's not a problem for a single use item, but once you get into things like medical linens or something like that, you don't want it washing out.
No.
And
Yeah.
that's for two reasons. It becomes less effective, but it also comes back to this issue that EPA is starting to raise for some of those metal ion based products. They are winding up in the wastewater stream.
Mm. Mm-hmm.
So this is the big problem for nano silver in kind of mass market applications where there might be a lot of product in general use. When EPA first looked at nano silver in I think around 2000 or whenever that first started coming on the market, their initial approach was, well, silver has forever been recognized as an antimicrobial. This is just another form of silver. We'll approve it. And then they started finding nanosilver particles all over, every sample they took in a river or ocean or lake. And they actually opened back up a data call in on nanosilver, and that is kind of still up in the air what the long-term future is for that, because there's no way to capture those products in the wastewater treatment system. It's just too small. It's going to go right through.
Let me wrap with first. Thank you, Kerry.
Thanks, Gary.
One of the things, as I said at the start of the program today, was I wanted today to be a chance for you to talk with folks who typically wouldn't hear your story, perhaps have them say, did you think of this? Did you think of that? Let's close, Kevin, with what are, in your view right now, the three biggest challenges you have, or opportunities, or outstanding questions, or if anyone on the call knows someone who, that kind of thing, so we can best as a group help you. What would you ask for?
I think for us, I actually, I was kind of anticipating the question that came from Dwayne. I know the biggest challenge on our side to move to the next stage of commercialization is to answer some of those basic questions on biocompatibility. But really, the reason I wanted to come on is to find out, or get ideas from other people on some other applications and how to move it forward, because there are a lot of potential applications, and we certainly can't think of all of them, and I certainly can't figure out on most of them what the regulatory pathway may be.
Mm-hmm. Okay, well, I know you're talking with Michelle.
Yep.
So she can help you with that.
So it's interesting though, because I had a good call with Michelle a week before last, and I need to have a follow up with her, because I kind of took what I got in the conversation with her, and she gave me some feedback based on another project she had worked on, and I took that and I went to a couple of other people in the industry that are working on similar products right now, and this comes back again to maybe where our benefit is, because she had seen some significant challenges on a project where somebody was working with silver-based technology, and she told me kind of what they went through, and the feedback I got from a couple of other people said, well, the reason that product had such a problem with the regulators was because it was silver. So we think, I feel actually, when I talked to her, quite frankly, after I got off the phone, I was really, really worried, because she started telling me what those other people were going to have to spend if they wanted to get that product out there. But it may have been unique to that product, which I think helps us from a competitive standpoint.
Okay, and you might see, I won't elevate Jim right now because we're wrapping up. He's asking a question about if you've talked to military folks who have decontamination requirements. You already said you had trouble getting in to military conversations.
Yeah, I've actually, like I said, we've had, I just haven't been able to get to the right people. So as I said in earlier in the presentation, the guy that did some of the early work on this was at University of Georgia.
He's
and
not-
worked pretty closely with, or actually Georgia Tech, I should say, worked pretty closely with CDC and the Emory University Department of Infectious Diseases. And one of the people that looked at it there basically said, based on how we work on everything else, it actually should work pretty well even against things like Ebola. Question, we certainly can't test against Ebola ourselves. And I would be interested to talk to somebody again for a military type application. I just, I worked my network the best I could earlier this year, people that I thought could get me in, and I really just didn't have a heck of a lot of luck. So
I could bring Jim on. He had an idea. Jim, unmute. Unmute yourself.
Yep.
Okay.
Hi, Kevin. Yeah, back when we first put our compressor into production and they brought me in, the first thing I did was search the congressional budget for programs that were funding electronics integration.
yeah,
And that's where you find the big pots. The larger the pot of money, the more people are fighting for it. And what we did is we found the primes that were on those programs in the development phase.
Yeah.
And that part is key. And then got in with them. You may find, if you search the congressional budget record that comes out and you look for those R&D slated, it's the flavor of money that matters to get in. So you get in during the research phase.
yeah,
And then find the primes. And it's just an idea. You may have tried it, but it's a way to at least find who in the government and who the primes might be that would need what you have.
yeah. Okay, I appreciate that.
Thank you, Jim.
Yep.
What else? I had asked you for 3. If there's anything else that you'd like to ask of the group, Kevin.
Nope. Nope. I got pretty good feedback out of this, and I appreciate it.
My pleasure. Next week is Jim. We're going to talk about cooling, and I'm just glad to be able to make the network available to you so we can help get your product out there.
Hopefully, we're working on it.
Thank you everyone for joining us, and we will see you next week.
All right. Thanks a lot, everybody.
Thank you, Kevin.